It may look a little odd. It's split into two distinct wings, and this is for good reason. We're actually made up of NHS and also University of Oxford, and we're all under one roof, which is great because it means that the patients that are coming in to see their specialist doctors or nurses to address their diabetes or their hormonal conditions are actually in the same place as the people that are conducting the research into the biology behind those diseases. So we've got this nice patient area here in the top right-hand corner, and we actually as scientists walk through this every day. So we're kind of constantly reminded why we're doing our job, which is really great and really important. And another thing that we're really keen to do in Opdem is to kind of interact with our patients and also feedback as to where their charity money goes, what we're doing to make a difference. And you can see here from the bottom three pictures that we recently had a public engagement day where we invited patients and any members of the general public to come in and learn a little more about what we do. This looks like a rather scary man here. We don't do anything crazy. This is just someone who works within our island isolation facility. So this is part of the Department of Surgery that prepares pancreas and pancreas tissue for transplant to patients. Gosh, you can see my script. We should refrain from using that.
Let me put your slides on.
Oh, thank you. So, who am I? Well, I work within the teams of Professors McCarthy and Browning, who are in this top right-hand corner here. And our group is actually made up of lots of different types of researchers. So we have clinicians, we have research nurses, we have computational biologists, and we have laboratory-based researchers. And as you can tell from this picture of me, I'm actually in a lab quite a lot of the time, so that's where I fit in that kind of bigger picture of research. And our interest is actually in the genetic causes of diabetes, so what happens in a patient's DNA that makes them have a disease. So we use these findings to try and understand what makes patient cells different and to behave in a way that we may not expect. And we try and use this knowledge to aid the design of new drugs and treatment strategies that can then be passed on to our patients. And the reason that we're working with Tim and Margosha is that because we're very keen to understand why diabetes occurs in patients with Wolfram syndrome. Thank you. So how does this work fit into our laboratory? Well, we're able to study Wolfram syndrome through a big European Union funded project called STEMBank. So this is a huge collaboration of about 23 different universities and about 15 or so drug companies that span Europe. And together we are studying cells from over 500 patients. So this doesn't just include diabetes and people with Wolfram syndrome. This spans a whole host of disorders, so things ranging from migraine to Alzheimer's to bipolar and so on. So our ultimate aim is to generate a resource of cells from a variety of patients that is then available to all researchers, which will help us make better disease models, help develop new drugs, and most importantly, help us make safe drugs. So, what does my work specifically involve? Well, it's leading on from what Professor Arano spoke about this morning. Got tech gremlins clearly.
How it's gone? Is it plugged in the wall there? Yeah. Plugged in. Sorry about that.
It's not just me. So my work involves changing your skin cells into a special type of stem cell. And the reason we do this is because stem cells can then be turned into any type of cell that makes up a human body. So as you can see from this diagram here, we have nice stem cells, which we can then use to make streaked bacon or muscle, some kidneys, lung, more skin cells, pigment cells that make up moles. So we really do have a kind of endless range of possibilities of cell types we can then study. And this is really important because what we actually want to do is look at the tissues that are affected in Wolfram syndrome. So these are largely nerves or neurons. I'm going to press this and hope it works. I normally embarrass myself, but the computer's helping me. Yeah, I must be lucky. Okay, so bear with me. That's all right. So yes, so we make nerves or neurons, which very surprisingly actually come from the same developmental pathway that skin cells are made from. Or we can make pancreas cells, which is the tissue that's affected in diabetes. And these actually come from the same pathway as our lungs and things like our thyroid gland. So how do we make a stem cell? Well, the first step is to take a skin biopsy. So as you can see from this photograph here, our clinicians or our research nurses use a special skin tool which basically either slices or makes a kind of hole punch in the skin. This picture here, picture B, makes it look huge, and you can see that there's lots and lots of layers of skin cells, but these are actually maximally only 4 millimeter deep, so less than half a centimeter, which, if you want a comparison, is actually the size of a match head, so it's really, really tiny, and we can get all of the material that we need from this small incision. And there's also, I discovered yesterday, a cool gecko that's the size of a match head. So next time you're having your skin biopsy, you can picture that. So once we have our skin cells, the next thing we need to do is expand and grow them. And the way we do that in the lab is we chop up the skin biopsy into much smaller chunks. We plate them in a special dish with a broth which has lots of nutrients and things to help the cells grow and replicate. And once we've done that, we can look under the microscope and see that we have a nice big population of skin cells. So these are called fibroblasts, and it's this that we want to take through to our next stage, which involves treating them with a special cocktail of chemicals. So the way to think about this is that our DNA is the kind of part of our cells which forms a recipe book, and it dictates how that cell looks and how that cell behaves. And by adding our cocktail of chemicals, what we do is we change the genes that are expressed so that are on and off in the cell, and we can actually make them look and behave like a different type of cell. So with this special formula, which Professor Arano mentioned, gained the Nobel Prize 2 years ago, we can actually change something that behaves in the skin cell into some stem cells. So that's really revolutionized us and our work as biologists. So once we've made these stem cells, as you may imagine, because they were originally something else, they're not very happy. So we have to do lots and lots of checks and balances along the way to make sure that they truly do remain as stem cells. And what I wanted to show you here was the type of experiments that we do to ensure this. So what I've done here is I've used special fluorescent molecules to label different molecules within the cells that make them behave as a stem cell. And then I'm able to look under the microscope and make sure that every cell in the dish has markers of stem cells in them. So here's stem cell gene 1, 2, and 3, and as you can see, they all play very nicely. However, if I applied these same fluorescent molecules to skin cells, we wouldn't see anything at all. So we can do lots of things like this along the way to make sure we definitely know what we've got. So in our laboratory, because we're a genetics diabetes lab, once we've got these stem cells, we're actually turning them into cells of the pancreas. So again, this is your organ that may not be behaving normally if you suffer from diabetes. And again, we label different molecules within the cell to make sure that they're the right thing, nice fluorescent markers. And what we've been able to do is take our stem cell, which has got nice red stem cell genes, and again add a new cocktail of chemicals, which makes them light up when we know that they've made pancreas. So to walk you through these images, these are the types of things that I see in the laboratory. I can add a special dye which stains all of the cells in my dish blue. I can add different ones which then pick up things that we know make up a pancreas cell. And when I combine all the images together, I can see that I get this nice yellow colour, which means that every cell is now no longer a nice red stem cell, it's actually behaving like a pancreas. And what we've been able to do is take these cells all the way down into special ones that express things like insulin and other hormones that we know are important in diabetes. And all of a sudden we have a system where we can really study what changes in patient cells in the context of hormones that we know may play a role. So, finally, just to touch on some other experiments that I'm doing. So as a geneticist, we're interested in utilising some of the new tools that Professor Urano mentioned, which allow you to effectively chop and change the DNA within a cell. So these are a bit like molecular scissors that allow you to kind of introduce a mutation or change a mutation or completely remove a gene in its entirety. And actually, what we want to do when we do experiments like that is to look to see if we can change the mutations and if we can get improved cell function once we take that away. And this helps us to understand better the changes and the things that go wrong in Wolfram syndrome patient cells. And not only this, but this provides an opportunity to test molecules for new drugs and new ways of treating the disorder, which we'll discuss so elegantly this morning. So, you've already seen this picture today, but Tim, if you wouldn't mind clicking again. I think he looks rather grumpy, so he's got a smiling face there. But your kind donation of a skin biopsy helps us understand what happens in cells from patients with Wolfram syndrome. And I really do want to stress that together we can find new ways to treat patients. So whilst I'm in the lab doing this every day, there is no way I could do any of my work without your kind involvement and interest and willingness to donate such valuable tissue. So really, I cannot stress enough that it's really important and it really does make a difference. So thank you to everyone that's involved, most importantly the patients, family and healthy volunteers that donate their tissue, everyone at the University of Oxford, so Professors McCarthy and Groyne, who are my boss, and other members of the team. Huge thanks to the University of Birmingham, Professor Barrett and Dr. Zeitka, who were the kind of real leads in this project initially, because they're the link to you guys and they're the ones that have this vision for treatment and know what makes a difference, and it's just great to be asked to be involved, so thank you. Other members of STEM Bank, the funding bodies, also Eurowar, who we're not part of directly, but I know Tim is, and who are responsible for the tissue collection in the first place. So thank you, and if you have any questions, please do ask.